Good evening, all. Welcome to yet another session of Cell, the Unit of Life, lecture number 2 from Biopoint Stream the World of Bio, and it's a premiering on YouTube. Okay, guys, we have completed the prokaryotic cells, and now today we are going to learn or discuss about some of the organelles of the eukaryotic cell. Clear with that, and we are moving forward in today's session. Yes, guys, we are discussing with the chapter Cell, the Unit of Life. And now we are having the second lecture of this chapter. And those who are here without watching the first lecture, please just watch the first lecture of this chapter and come to view this second lecture. And now, as I have told you, we are going to discuss the eukaryotic cell and some of its organelles. Okay, clear with that. So then let's move forward and hope you all are fine and you all are attending the exams well and perfect and fine with your marks. Okay, okay, guys, and I'm after a long time I'm back to take class for you and just move forward with the first topic of the day, the eukaryotic cells. So what we have to discuss from this topic, what is there to learn? Let's take it. So what is a eukaryotic cell? They have membrane-bound nucleus and organelles. So membrane is present around the nucleus as well as the organelles. Whereas in case of prokaryotic cells, they have an incipient nucleus or a nucleoid. That is, there doesn't have a boundary or there doesn't have a membrane to bound their nucleus. Okay, so the presence of membranes give clear compartmentalization of the cytoplasm. They have complex locomotory and cytoskeletal structures. Okay, they have complex locomotory and cytoskeletal structures, and the genetic material is organized into chromosomes. The genetic material is organized into chromosomes. Clear with that? Cell organelles in eukaryotic cells. So which are the organelles that you have to study? Cell membrane, cell wall, and the endomembrane system. That is the GERL system. And it's an AIMS question. What does GERL indicate? GERL refers to Golgi, Endoplasmic Reticulum, Lysosome. And then what about this vacuole? They also help in that their function. Okay, GRL system performs a specific function and they too help in that, and which we will learn in the coming slide. Okay, so vacuole is also associated with them, but it is not written in this word. Okay, so it's a what previous year AIMS question. GERL indicate what? Golgi, not Golgi apparatus, Golgi, endoplasmic reticulum, and lysosomes. Mitochondria, plastid, ribosomes, cytoskeleton, cilia and flagella, centrosome and centriole, nucleus and microbodies. Hope today till vacuoles till here we should complete if time permits. Clear with that? So in the first cell, this one is the first cell which you are, and this is the second cell. First one is the plant cell and second one is the animal cell. So first we are going to start with the topic of cell membrane. So cell membrane, as I have told you, is also known as in the chapter of which chapter I have discussed that, I think in the chapter, yeah, this chapter right at the beginning itself. Okay, so cell membrane, as I have discussed, it is also known as plasma membrane. Oh my God, I'm not able to write properly, that's why. Just wait. Okay, as I have discussed, it is also known as what? Plasma membrane. Okay, biomembrane, plasmolemma. Okay, plasmolemma is also another term for cell membrane. Plasmolemma. Oh my God. Okay, clear with that. So chemical studies on human heart BCs show that the cell membrane is lipid bilayer, protein and carbohydrate. Next slide, you can see this have a clear diagram. Okay, lipids mainly phospho. So All these are things that you have to learn very carefully. Okay, okay, for that. So the lipid bilayer, okay, the lipid bilayer is purely composed of phosphoglycerides. Phosphoglycerides, or it is sorry, the yeah, exactly, the lipid bilayer is composed of phospholipids, and they have an outer polar head and an inner hydrophobic tail. Okay, like this, their structure is they have an outer polar head and a non-polar tail. Clear with that? So the non-polar tail of the saturated hydrocarbon is protected from the aqueous environment. Okay. And their head, polar head is hydrophilic and their non-polar tail is hydrophobic. Okay. Polar head is hydrophilic and polar non-polar tail is hydrophobic. So since the tail is hydrophobic, then how does the water enters the cell? Because they have certain structures in their cell known as aquaporins. Aquaporins. This aquaporins allows or permits the entry of water into the cell membrane. Okay, clear with that? And therefore, due to the presence of this aquaporins, those cell, what the non-polar tail of the lipid bilayer is hydrophobic, water enters the cell membrane. Clear with that? Ratio of protein and lipids varies in different cells. In human RBC, there is 52% protein and 40% lipids. So in case of human RBC, the normal count is 50 to 52%, 50 to 52% protein, 40% or 50 to 50% protein, 40% lipid, and 8 to 10% remaining 8 to 10% carbohydrates or carbohydrates, cholesterol, etc., etc. Okay, so based on the extraction membrane proteins are of two types. Okay, integral protein and peripheral protein. Integral protein and peripheral protein. So look, this is the integral protein and this is one is known as the peripheral protein. So what is this integral protein? This integral protein is also known as intrinsic protein. Intrinsic protein. Clear with that? So this integral protein partially or totally buried in membrane. Okay, and peripheral protein lie on the surface of membrane, that we will discuss. Okay, and this peripheral protein, since integral protein is known as intrinsic protein, then peripheral protein is known as extrinsic protein. Clear with that? And this integral protein is also known as channel protein because it acts as a channel. Okay, it acts as a channel, and therefore this is known as channel protein. And this is what we say to be the fluid mosaic model. Okay, fluid mosaic model, and it is universally accepted, I mean, yeah, well, what universally accepted and proposed by Singer and Nicholson in 1972. Okay, it is the universally accepted model, fluid mosaic model, proposed by Singer and Nicholson in 1972. And this model looks like protein icebergs in a lipid sea because it is a lipid bilayer. Okay, clear with the intrinsic and extrinsic protein and what we were discussing. With yeah, because in this lipid bilayer, this is a lipid bilayer, right? So in this lipid bilayer, these proteins are just embedded. So it looks like the protein icebergs in a vast lipid sea. Clear with that? So this is the fluid mosaic model proposed by the Singer and Nicholson in 1972. According to this, the quasi-fluid nature of lipid. What is this quasi-fluid? Quasi-fluid means semi-fluid, that is somewhat jelly-like. Nature of liquid enables lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. Okay. So the quasi-fluid in nature, this shows that the cell membrane is quasi-fluid in nature, that is semi-fluid or jelly-like. Okay, clear with that? And this feature of cell membrane is universally accepted. Okay, for example, from what in case from chlamydomonas to the cell of ostrich, that is from monerans till humans or till animalia, we can see the common feature of cell membrane. So universal feature. Clear with that? So then what is their function? Their first function is the transportation. Okay, that is transportation can be of two types. That is, there is active transport and passive transport. The second function which they have is they are quasi-fluid in nature. That is one of the property or its function. Okay, third one, it is that they show flip-flop mechanism or transverse diffusion. They show transverse diffusion. It is one of the function of this cell membrane. We said that the cell membrane has aquaporin, which allows the entry of water, whereas in case of cell wall, the structure known as porins is present, and it is seen in prokaryotes. In most of the prokaryotes, mitochondria, we have this cell wall of mitochondria, we have this porins, and in case of chloroplast also we have porins. Clear with that? So types of transport which we discuss is the passive transport and the active transport. Passive transport refers to the common diffusion and osmosis, that is without using any energy, and active transport using energy against the concentration gradient. So passive transport is the movement of molecules across the membrane along the concentration gradient without expenditure of energy. It is of two types, that is a simple diffusion and osmosis. Simple diffusion means the movement of neutral solute across the membrane. Osmosis, the movement of water by diffusion across the membrane. Polar molecules cannot pass through the non-polar lipid bilayer, so they require membrane carrier protein for their transport. That is what we said to be the facilitated diffusion. Clear with that? And further classification of this osmosis as endosmosis and exosmosis, and all these topics we have there in the chapter of transport and plants. Clear with that? So this is the active transport by using the energy against the concentration gradient. So next structure that we are going to learn is the cell wall. It is a non-living rigid structure formed outer to plasma membrane of fungi and plants. Cell wall of algae is made up of cellulose, galactans, mannans and minerals like CaCO3. In other plants, it consists of cellulose, hemicellulose, pectins and proteins. So composition of cell wall is different to different organisms. For example, the cell wall of monerans, it's made up of peptidoglycan. The cell wall of dinoflagellates, it's made up of cellulose and chitin. Okay, cell wall of um, okay, then cell wall of diatoms, it's made up of leucosin. I hope so. So cell wall of young plant cell, that is the primary cell wall, is capable of growth. It gradually diminishes as the cell matures, and secondary cell wall is formed on the inner side, that is towards the membrane. Middle lamella is a layer mainly of calcium pectin. It glues the different neighboring cells together. The cell wall of middle lamellae may be traversed by plasmodesmata, which connect the cytoplasm of neighboring cells. All these things are which, what not much important. The other thing which you have is the primary cell wall composition, which is made up of cellulose, and the secondary cell wall is made up of lignin and suberin. Secondary cell wall is made up of lignin. Hope you can see. I can't talk with you since it's a premiering session. And suberin. Okay. Then what we discuss in case of this plants, what cell wall and what semi-permeable, what we can say about that? We can say that this cell membrane is selectively permeable, not semi-permeable. It is selectively permeable, and cell wall is freely permeable. Okay, what is the function of cell wall? It gives shape to the cell, protects the cell from mechanical damage and infection, helps in cell to cell interaction, act as a barrier to undesirable macromolecules. Clear with that? So what you should do is that just read. Only basics are just there from these parts. That's why I'm not going into the minute details. Next, we have the endomembrane system. Endomembrane system is a system including endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. Okay, that is a GERL system. Okay, this system performs a common function known as protein trafficking or protein targeting. Okay, protein trafficking or protein targeting, because the protein prepared by the ribosomes is just traveling from, or is just passing from how you pass a ball, like it passing from endoplasmic reticulum to Golgi complex to lysosomes, then to lysosomes to vacuoles, like that. Okay, therefore this is what we say to be the endomembrane system. Then what about endoplasmic reticulum? It is a structural or the supportive framework of a cell. Okay, so this is divided into this divides intercellular space into two components, that is the luminal and the extraluminal, that is the inside endoplasmic reticulum part and the cytoplasm. And this endoplasmic reticulum is of two types, that is the RER and the SER, rough endoplasmic reticulum and smooth endoplasmic reticulum. So how this rough and smooth endoplasmic reticulum are formed? If ribosomes are present on the surface of a endoplasmic reticulum, it is known as the rough endoplasmic reticulum. And if the ribosomes are detached from their surface, then we can say it is a smooth endoplasmic reticulum. Clear with that? So they frequently have the ribosomes on this structure and is absorbed in the cells, actively involved in protein synthesis and secretion. They are extensive and continuous with outer membrane of nucleus. Smooth endoplasmic reticulum ribosomes are absent, major site of synthesis of lipids. In adult cells, lipids like steroidal hormones are synthesized in smooth endoplasmic reticulum. Okay, that's all for this endoplasmic reticulum too. Nothing more to learn from this topic. And this endoplasmic reticulum we can see in three forms. That is in the form of vesicles, second one in the form of tubules, third one in the form of cisternae. Okay, these are the three forms in which we can see this endoplasmic reticulum. Clear with that? From nuclear membrane, the after what ribosomes are produced in the rough endoplasmic reticulum, which is passed on to the Golgi, and then it is passed off to lysosomes. Okay, so by blobbing of rough endoplasmic reticulum, we can get smooth endoplasmic reticulum. Okay, and what is their function? Their function is to detoxification of drugs and specialized for synthesis of lipids and steroids. For any doubts, just comment in the chat box, and I am reading your chats in this premiering section too. Okay, next we have the Golgi bodies. Golgi bodies. Okay, so what about this? What can we say about this? They are we cannot say it as Golgi apparatus or Golgi bodies. That's what we have to discuss very first. So we should say Golgi only, because what is then this Golgi body and Golgi apparatus? Golgi bodies are something, or this Golgi found in protozoans and some fungi, and Golgi apparatus are found in eukaryotes. And now we are discussing cell organelles of eukaryotes, so we can say it as Golgi apparatus, but commonly it is Golgi. Okay. And this Golgi apparatus, in case of plants, it is known as dictyosomes. All of you know, right? All these things. In your 9th standard, it is there. CBSE 9th standard or state 9th standard, everyone, everywhere it is there in 9th. Michael, I'm not able to write. I don't know what is happening to this. Okay, just leave it. Okay, so then 4 to 8 cisternae are observed. So they consist of flat disc-shaped sac, and that is known as the cisternae. Look, this is the cisternae, and there are 4 to 8 cisternae in a Golgi. Clear with that? Okay, so they synthesize glycoproteins and glycolipids. Okay, they act as a major post office of the cell, that is a storing and packaging. They secrete their certain enzymes. Okay, so they are known as a secretory vesicles too. Clear with that? And this cisternae are concentrically arranged with convex face and concave trans face. Okay, cis and trans faces are totally different, but they are interconnected. Clear with that? And their function, as we have discussed, formation of glycoproteins and glycolipids, glycosylation of proteins and lipids, secretes materials into the intracellular targets or outside the cell. Materials to be packed as vesicles form the endoplasmic reticulum fuse and with the cis phase and moves towards the trans phase. All these are not needed. Okay, not needed, not needed, not needed. Okay. This is the lysosome. They are membrane bound. What is lysosome? They are the suicide bags of the cell. They are highly acidic in nature. That is, they have a pH of 4.6 to 5. All these are not required, which is given here. Okay, they are active at the acidic pH. Okay, consists of 50 different types of enzyme. What we can say if we consist of 50 different type of anything like that, this lysosome consists of 50 different types of enzymes to just digest the foreign particles or anything that would affects the lifespan of that cell. Clear with that? And they shows polymorphism, that is for one phase it can be seen at one time in what for example, if it is a prime, it will be a primary lysosome at one time, and if you look next time, it will be a secondary lysosome. After that, if you look a third time, it will be a residual lysosome, and if you look fourth time, it will be an autophagic lysosome. All these are the different forms of this lysosomes which we have discussed. Okay, nothing more in that. That all are not at all needed. Clear with that? Next, we have the vacuoles. I think that's all for lysosomes. Yes. Next, we have the vacuoles. They have a boundary known as tonoplast. They contains water, sap, excretory product, and other waste which are not useful for the cell. In plant cells, 90% of the volume of the cell we have vacuoles, and they are the membrane-bound space found in the cytoplasm. And these vacuoles can be of three types: contractile vacuole, food vacuole, and gas vacuole, and nothing in detail. Contractile vacuole found in amoeba, food vacuole in some protists, gas vacuole in blue-green algae, purple and sulfur bacter. Clear with that? Look, tonoplast facilitate the transport of ions and other materials against the concentration gradient into the vacuole. Amoeba contractile vacuole for excretion. Food vacuoles are formed in some protists, formed by engulfing the food particle. That's all, and thank you for your patient listening. And that's all for today. Hope I was too fast in today's session because it was a premiere. I cannot video chats and interact with you. That's why I just moved a bit fast. Okay, and I'm having a short of time now. Okay, that's why I'm not taking live for you. Clear with that? And it's me, therefore, living from your favorite and my favorite biopoint stream, the world of bio. Bye bye.